Imagine standing in a world where an entire universe exists around you, but you cannot see it. The water you drink looks perfectly clear. The dust floating through a beam of sunlight appears harmless. A drop of blood is simply red. A piece of cork is just cork. You have no idea that beneath the limit of your eyesight, countless tiny structures are moving, dividing, feeding, and dying. For most of human history, the microscopic world did not exist, not because it wasn't there, but because nobody could see it. Then, sometime around the beginning of the 17th century, humans began building an instrument that would change that forever: the microscope. The earliest compound microscopes appeared in Europe around the late 1500s and early 1600s. Their exact inventor is uncertain, although Dutch spectacle makers Hans and Zacharias Janssen are often credited with early versions. These first instruments were little more than tubes containing several lenses. They were crude. They produced distorted images. But they possessed an extraordinary ability. They could make the invisible visible. And once people learned how to look closely, the familiar world suddenly became strange. A flea was no longer just a tiny pest. Under magnification, it became a bizarre armored creature covered in spines and delicate structures. A louse looked almost monstrous. One of the first people to truly exploit this new world was the English scientist Robert Hooke. In 1665, Hooke published a remarkable book called Micrographia. It contained enormous illustrations of things he had examined through his microscope: fleas, lice, insects, plants, hair, crystals, and even the tip of a needle. The pictures were astonishing. Readers could suddenly see details that were invisible in everyday life. But Hooke made one observation that would become especially famous. He placed a thin piece of cork beneath his microscope. Instead of seeing a solid piece of material, he saw a honeycomb of tiny compartments. They reminded him of the small rooms, or cells, occupied by monks. So he called them cells. Hooke was not actually looking at living cells as we understand them today. The cork was dead plant tissue, and what he saw were the remaining walls of the cells. But the name survived, and eventually, cell would become one of the most important words in biology. Yet the microscope still had a problem. The instruments of the time were limited by the quality of their lenses. Then came a remarkable man from the Netherlands. His name was Antony van Leeuwenhoek. He was not a university professor. He wasn't a famous physician. He was a tradesman who became fascinated by lenses, and he became extraordinarily good at making them. Instead of relying on several imperfect lenses, Leeuwenhoek developed tiny, powerful, single-lens microscopes. Some were only a few centimeters long, but their magnification and clarity were astonishing for the time. And Leeuwenhoek began examining almost anything he could put beneath a lens. Water, blood, plant material, scrapings from his own teeth. And what he saw shocked him. In droplets of seemingly ordinary water, he discovered tiny living organisms moving through the liquid. He called them animalcules. Today, we know many of them as bacteria and protozoa. Think about what that meant. For thousands of years, humans had looked at a puddle of water and seen nothing unusual. Leeuwenhoek looked again and discovered that it was alive. A drop of water had become a crowded landscape. Tiny organisms swam through it. Some spun, some darted, some moved slowly through the liquid. And suddenly, the world was revealed to be vastly more crowded and complicated than anyone had imagined. But perhaps the most important discovery wasn't any single organism. It was the realization that life had an entire hidden scale. There was a world beneath the world, a world too small for the human eye, yet filled with extraordinary complexity. and that realization would eventually transform medicine and biology. Scientists would discover that plants and animals are made from cells. They would learn that microorganisms exist almost everywhere. They would eventually connect microscopic organisms to fermentation and disease. And centuries later, increasingly powerful microscopes would allow humanity to see chromosomes, viruses, proteins, and structures inside individual cells. The microscope did more than magnify objects. It changed the scale of human curiosity. Before it, we looked upward at the stars and wondered what existed beyond Earth. After it, we could look downward and discover another universe hiding beneath our fingertips. A drop of water became an ecosystem. A piece of cork became a landscape. A speck of dust became a collection of strange particles. And an ordinary human body became a vast community of microscopic structures and organisms. The remarkable thing is that none of these worlds had suddenly appeared. They had always been there. The bacteria had always been swimming. The cells had always been there. And perhaps that is the true origin story of the microscope. It wasn't the invention of a machine that made the invisible world exist. It was the invention of a way to discover that the invisible world had been there all along.